Hi, this is Mr. Young, and in this video podcast we focus on protein structure and function. As we look at proteins, we find that they are among the body's most diverse macromolecules. Truly about 50% of your body's organic matter comes in the form of proteins. But not just in humans do we see proteins, we see them all over the place. Here, for example, we see what's thought to be the most abundant protein on Earth, this protein called rubisco, which plays a role in the fixation of atmospheric carbon dioxide into a form that can be not only generated and made by plants, that being glucose, but also passed along through the food chain. In other organisms, we see proteins like you identify here. This is VP40, which has been the topic of much research this past year as I've had scientists investigating the pathogenesis of one of the deadliest viruses known to man, that being the Ebola virus, which in some cases reaches mortality rates of 90%. Through understanding the viral matrix protein shown here and how it allows the Ebola virus to bud, for example, and to spread from one cell to another host cell, through understanding the structure and function of the VP40, scientists are hoping to better understand how to combat it in terms of not just treatment but also prevention of infection. Finally, below you see the number one in terms of abundance protein in mammals, this being the protein called collagen, which is about 1/3 the total protein content in the human body. Collagen is found everywhere, from dermal fibers that make your skin flexible and yet strong, and in your bone and in your tendons and your ligaments. Collagen is a primary fiber, primary protein. Bottom line is the shape of our proteins will have everything to do with what they do. And so as we look at our proteins today, let's look closely at the shape and how the shape is influenced by the smaller building blocks that build proteins. And that's what we see right here. Just as carbohydrates were built from simple monomers that could be put together to build a polymer, from simple carbohydrates to complex carbohydrates, here we see how the simple monomers for proteins are formed, and we call these amino acids. So there are 20 regularly occurring amino acids across all taxa, with a couple that show up in a few specific species apart from those 20. But as you look at all these amino acids, what stands out as far as what is in common among them all? Good. You've identified that each one has this NH3 group sticking off on the left side as it is on these different molecules. We happen to call this an amine group or an amino group. And then in addition to that, you saw the similarity they all possess in this functional group coming off. This functional group, if you recall, is referred to as a carboxylic acid. And so if you put those two groups together, what we find is an amino acid. There you go. This is how we came up with the term amino acid. In addition to those similarities, we see a number of differences, but the primary one is just the fact that there are a variety of different R groups. The R group is really what defines the amino acid, and those R groups will confer various properties. For example, all of these R groups result in non-polar amino acids, as you can see over here on the left. A lot of symmetry down below here, not many oxygens sticking off that have lone pairs of electrons, but non-polar. Polar, notice the presence of oxygen that you now see, or sulfur, nitrogens that are hanging off down below off of the end of this R group, resulting in a polar molecule. And finally, electrically charged, where you see positive or negative charges. Here are the positive charges here, negative charges on the far left here, and that will lead to also to some acidic versus some basic properties in these various amino acids. So for your studies, be sure to review the carboxylic acid end versus the amino end to identify how those amino acids are actually formed. Well, how do amino acids join together to build more complex molecules, like proteins? Well, here we see amino acid number 1 with its adjacent amino acid number 2, and we're curious about how they form a bond with one another. You identify here the location where the bond will be off of the carboxylic acid end of our first amino acid, off of the amino end of this amino acid. And when the bond forms shown here, we refer to this as a peptide bond. And that peptide bond connects one amino acid to the next, and that's why we call this a dipeptide, or two peptides. In reality, this will continue and work in this direction to form multiple other peptide bonds until we have not just a dipeptide, but a polypeptide. And so we're using monomers plus monomers to build polymers, and in the process of doing that, just like we saw in the production of complex carbohydrates, we end up with a condensation reaction. And it's a condensation reaction because of the fact that liquid's lost. We've lost a water molecule. This is dehydration synthesis, very good, and it's happening again. Well, as we look at the variety of structures within proteins, we're going to break it down, starting with its primary structure, which is simply representing how the variety of amino acids are joined together through peptide bonds, or the sequence of amino acids. And remember that these amino acids are in their order because of the recipe that codes for that order, and that recipe happens to be DNA. The central dogma of all molecular biology is that DNA is the recipe that is used to build protein, after all. And DNA is formed in specific codes that code for unique amino acids. And those amino acids, when put in sequence, provide what we call the primary structure of a protein. As you look at the protein's primary structure, you may wonder, well, big deal, what the sequence of amino acids is. One example where it's a huge deal. Here you see the amino acid glutamine that exists as the sixth amino acid in a sequence of 146 different amino acids that comprise to form the hemoglobin molecule in humans, a molecule which is responsible for carrying oxygen in red blood cells. That molecule, unfortunately, in the right sequence, is shown to have a different sixth amino acid, in this case valine. And that single amino acid change out of all 146 results in a change in the morphology of the cell. Look at these shapes of cells related to the normal red blood cell biconcave disc. Which one do you think will have a difficult time getting through capillaries as blood cells line up in single file line? Yeah, you got it, sickle cell anemia. And so the whole reason why this forms is because of that amino acid here. The glutamine is a hydrophilic amino acid, meaning that it is a lover of water, and that will result in a bending of that portion of this molecule out toward the outside of the overall protein, toward the aqueous environment. Whereas over here, the valine is a hydrophobic amino acid, which means that it's going to result in a bending inward of that protein for the hemoglobin. And the net result is you end up with the morphology that you see here. Primary structure absolutely matters. Well, from primary structure, we see secondary structure builds on the complexity of a protein. Here we see that hydrogen bonds between adjacent amino acids can result in a variety of different formations. And the two shown here are what we refer to as alpha helices, or the ribbon shapes that you'll see in these various models, as well as beta pleated sheets, these folded sheets that you can see here on the right hand side. You may wonder, well, why does those matter, the secondary structure? Well, ask Peter Augury, the 2003 Nobel Prize in Chemistry award winner, who discovered the aquaporin channel protein, a protein embedded within the phospholipid bilayer of cells, allowing higher rates of water transport across cell membranes than was previously seen just through the phospholipid bilayer. But his discovery of the aquaporin and its 6 alpha helices were critical to him winning that Nobel Prize in 2003. Excuse me. So secondary structure certainly matters. Tertiary structure takes a look at now the larger protein and the variety of different intermolecular attractions that can happen. For example, hydrogen bonds, yes, certainly we see those here, as well as among different R groups now, hydrophobic interactions, as you see two non-polar portions that are close to one another now. If you're working with cysteine or methionine, two amino acids that have sulfurs in their R group, we can see disulfide bridges forming. Or finally over here we see ionic bonds that form between oppositely charged portions in the R groups of these different amino acids. So the bottom line here, we're looking at interactions among R groups in the tertiary structure, whereas in the secondary structure, we're really looking at molecular interactions and intermolecular forces between adjacent amino acids and specifically their non-R group regions. Lastly, we see the quaternary structure, which relates to now all the different protein subunits that will combine to form our larger protein. Case in point here is a protein called hemoglobin. We saw this earlier with the sickle cell anemia case, but hemoglobin is comprised of 4 smaller subunits that gather together, and their foldings allow for them to interact and carry these heme molecules, which would be that site of attachment for oxygen. But the quaternary structure is really what allows this protein to have function in the end, whether it's hemoglobin or collagen with its rope-like structure here. The quaternary structure in the end is the culminating structure that gives proteins their function. So how do proteins lose their function? Well, if structure influences function, then changing of structure, as you can see here by the process of denaturation, will likewise change the function of our proteins. And that's exactly what happens in the presence of changing temperatures beyond normal range of tolerance for proteins, changing pH, as well as change in salinity. Any of these variables, as well as others, can result in a change ultimately in the structure of our protein, which can cause our proteins to lose function. A couple of examples for you to be looking for. Temperature. Next time you cook an egg, admire that egg white before you put it on the hot surface of your skillet. As you go from transparent egg whites to truly egg whites. How is that happening? Proteins are heating up. They are beginning to get denatured, and that process of denaturation will cause there to be that change in color. In addition to that, you can identify how the next time you have a fever, there is cause for concern if that fever gets really high because proteins within your body can get denatured. And so I hope this is a good look for you to see how protein structure relates to the amino acid sequence that then will lead to a variety of folding, whether it be in short segments or the secondary structure, larger segments of the overall protein, the tertiary structure, or a variety of different subunits that combine together to form the quaternary structure of proteins.